This is the United Podcast Network.
This is Voices in Validation, brought to you by IVT Network. IVT Network is your expert source for life science regulatory knowledge. Voices in Validation brings you the best in validation and compliance topics. We interview industry experts from pharma, biotech, med devices, and laboratories. Here is the host of Voices in Validation, Stacey.
Thank you, and welcome to another episode of Voices in Validation, brought to you by the IVT Network. Each week, we bring you timely interviews with life science experts and discussions focused on trending topics, frequently asked questions, and industry best practices. This week, we are pleased to welcome Dr. Ross Caputo from Eagle Analytical. It's great to have you here with us today, Dr. Caputo.
Well, thank you very much. It's my pleasure to be here.
We are looking forward to a discussion on contamination control and sterility assurance for compounding facilities. And just for a little background for some of our listeners, contamination control is very important for medical devices, pharmaceuticals, preparations, tissue materials, and any other materials that claim to be sterile or free from viable microorganisms. And any contamination that does occur must thoroughly be investigated, which is often time-consuming, causing potential delays in production, and this is true for compounding pharmacies of all types. So we really want to talk a lot today about controlling contamination and what can be done to assure, sterility in their facilities. Before we dive in on that topic, though, Dr. Caputo, I'd love it if you could kick us off by briefly updating our listeners on the current requirements under USP and the FDA for compounding pharmacies in regards to sterility and, of course, contamination.
Well, okay, that'd be my pleasure. I think sometimes there's some confusion when we talk about compounding pharmacies and the USP and the FDA requirements. As we know, that compounding pharmacies termed as a 503A in the law, which means a dispensed prescription-based product, is always, and I mean always, governed by the state boards of pharmacy.
Right.
The FDA only gets involved if there's an issue.
Right.
And I think that's the only thing. That's that knock on the door and hello, I'm here to help you is a bit scary. But the reality is you have to follow the state board rules. And the USP rules for 797, which is all about sterile product testing, is what the FDA will ask the questions of a pharmacy when they walk in the door. If a state board of pharmacists comes in, comes in, clearly they're understanding the requirements of 797 and their unique board requirements. Now, board requirements for sterility and many other things vary from state to state. We go from the very highest level of scrutiny and specificity, which mimics exactly a GMP FDA type response, to those state boards that perhaps require very little, if anything at all. I'm not going to discuss those or which states there are. Those pharmacists in those states already know where they are. But at the end of the day, what they're talking about is that you must prove with a test for sterility that your product is free from contamination in a compendial test. A compendial test is a reference test. It tells you what to do, how to do it, what your responses should look like or not look like, and therefore it's a reference standard. And the FDA will always look to see if you did a sterility test, if you perform something called method suitability. And method suitability has been the biggest bugaboo lately. You'll see people that have been doing a test, and they said that my test looked good. And then either frankly, the state board or the FDA will say, but excuse me, you didn't prove that it actually worked with your product. As we know, some products can be bacteriostatic or bactericidal. Well, method suitability indicates that you can test that product in the presence of inhibitory compound, neutralizing it, to make sure that you, in fact, are doing something that's valid. That when you say it's free from contamination, it actually is. It's not just leaping. But I will tell you that sterility and contamination are the number one issue and sometimes the only issue that the FDA gets involved in a compounding pharmacy. It becomes an issue of safety. And we shouldn't, and we'll talk a little bit later, I'm sure, about contamination and sterility are not always absolute to be able to be tested one way or the other.
Sure, right. And so we're talking about, when we're talking about compounding in this case, you're talking about 503As, right? As opposed to
Well,
the
503B is an outsourcing facility.
right.
Very similar rules.
But they are considered much more of akin to the manufacturer. So the FDA would
Yes.
have a little bit more authority here, right?
Yes.
Yeah.
But I think the only issue that I want everybody to hear is that the sterility test is the same for everybody.
Right,
Whether it's
exactly.
a compounding pharmacy, a hospital, outsourcing facility, a laboratory, it doesn't matter.
Perfect.
The same thing.
So I just wanted to make that distinction, which is
Sure.
why I mentioned the FDA at the beginning. But yes, but the 503As, they're definitely under the state boards. So to move on a little bit, I wanted to get your thoughts on what the most frequent cause of contamination is, because obviously you can do the different forms of testing and prove sterility, but ultimately I would think you would want to account for any sources of contamination up front, so you'd have less chance of having any issues. So in your expert opinion, what do you think the most frequent causes of contamination are?
The most frequent contamination of a clean room is us, people, human
Yep.
beings. We bring it in all the time. We bring it in on our body, our face, our hands. That's why it's supposed to get gowned up and drawn up. We also bring it in when we bring in the materials that you're going to use for a test or to produce a product, by not effectively decontaminating it or disinfecting it before you use it. It has to be transported in. Today, to do a sterility test in a clean room, the clean room is going to be HEPA filtered air. The actual hood is an ISO 5, which is clean and cleanest, 1 in 100, right? And then you have an ISO 7, ISO 8. All of these things give us a cascade environment where we in fact learn that the only way to manage something and keep it clean is to never get it dirty in the first place.
Yeah.
So we have
That's
cascading
what I
air
was
of
alluding
positive
to.
pressure.
Yeah.
What's that?
That's what I was alluding to in the beginning of that question. Is like, yeah, obviously we want to keep contamination out to the best of our ability.
Yeah, and that's true. So positive pressure in our clean rooms to push everything out, make sure that dust particles and other contaminating entities don't come in to the area, but get blown out of the area if something shows up. But we ourselves bring it in. We ourselves, human beings, bringing the product in, will bring it in. That is the major source. If you have contamination, that's where it comes from. We certify hoods, we certify rooms, we certify airflows. We take countless and countless tests to show that we have a clean environment. So how does the clean environment get dirty? We do it.
With people, right? And it'll be interesting. This is a quick aside, but it'll be interesting to see with all of the, with all of the different steps that people have had to take during this COVID pandemic, to either whether it's shortages of PPE or different cleaning agents, et cetera, that are hard to get. If there will be any impact, or if there has been, I should say, any impact to the sterility, to the typical sterility that one sees in a compounding facility based on that. I don't know if you have any insights yet, or if it's still too soon to tell, right? Because we're still in the midst of this pandemic, but I would think it must have had some sort of impact.
Well, I think it's impossible that it wouldn't have an impact.
Right.
The probability of it is just on the other side of the coin. The good news with decontaminating surfaces and rooms and walls and stuff, if you could buy bleach is the best thing. It's a sporicide. It cleans pretty much everything. It can be pretty caustic to walls and the stainless steel and things like that.
Sure.
But in a time like this pandemic, it's probably the better thing to do.
Right. Agreed. So let's talk about sterility testing. I think anybody who's in the compounding world, and probably many people who are in life sciences in general, are familiar with this. It is a widely used process, particularly in compounding, and can be very helpful when designing a validation process. But as you alluded to, sterility testing alone is not the only indicator of maybe contamination or a process failure. What are the limitations in your experience of sterility testing?
Well, to be honest with you, I find the sterility test has so many limitations. It's something that you have to do. I mean, if you understand that a sterility test that you can have a contamination rate of 10% and still be passing it 15% of the time. I mean, it's just unbelievable, the numbers. For those of us that have been in the validation world, we understand that the sterility test is a catastrophic measurement system. For a sterility test to fail, it's bad. You're not measuring probability numbers. Terminal sterilization and sterilization in general is measured about a probability that is less than 1 in a million of a contaminated unit. And I'm telling you the things that are 1 in 10 or 1 in 100 will pass routinely. So the level of significance is tremendous. Sterility test tells you if you can't pass a sterility test, there's a problem.
There's a major problem then.
There is a problem. We consider it catastrophic.
Sure. Okay.
You mentioned process validation and systems like that. Of course, sterility test has a role in there, as do media fills and process validation, as do all the metrics that you have to do. For years, individuals would look at sterilization processes, whether they be aseptic or terminal or gamma. They try to use things called parametric release, where you're measuring physical engineering parameters to prove if that they are attained real time, you have a high probability of producing a sterile product. Steam sterilization is one of the classic, right?
Right.
121.1 with the delivered heat of 15 minutes. This puppy's sterile. So that if you have measurements going on that you know that was delivered, then you know that was sterile. If you go and sterility test that, you would expect it to always pass. And frankly, if it failed, you go ahead and question the sterility tester. You wouldn't question the process. But that's where all the other things, and I think that you're very wise to bring into process validation and designing a validation process is multifaceted. There's no one thing. A passing sterility test is absolutely no substitute for validation, none whatsoever.
It would seem to me, because I would think there are different substances or different bacteria or contaminants that are going to be hard to either get rid of or hard to, maybe hard to test, right? Are there certain things that are difficult to filter? Or you mentioned, I think, growth inhibiting properties for some of them. So maybe you can provide a couple of examples of unique challenges for sterility testing due to those type of issues.
Well, I think routinely we have seen in the past where certain antibacterial agents and antibiotics have been contaminated with organisms that were spores or dormant that are not picked up in the sterility test because of the ongoing activity of the bactericidal agent or the preservative, frankly. When you have a preservative in a product, you must neutralize the preservative before you test it. Otherwise, you can have a function of just dormant spores or dormant bacteria or different strains of bacteria. But we should not lose track of that. I guess I forget as a microbiologist that these media do not pick up all the organisms.
Right.
They don't claim to do it. They don't mean to do it. They're not designed to do it. In fact, if you were trying to grow an injured organism, you would not use these organisms. They use these media. Use something else completely different. So these things are done to get the majority of basically a mesophilic aerobic organism, grows at a normal room temperature, not an extreme thermophile, not something that grows at lower temperatures. It's not designed to do that. The whole intent of a sterility test, and I don't know if a sterility test was first used, I think in 1936 or something. That was very different when it got put together. And then over time, it's morphed into what we have today. And there's been the sort of like face masks. There's been this discussion of what the science tells us and doesn't tell us. It used to be that if you did a membrane filtration test, the incubation period was 7 days. Okay.
Right.
And then only the aseptic process had to go 14 days. I didn't know that the bug could tell whether it was contaminated from aseptic or steam, but that's okay. And it had a lot to do with that. But we have harmonization today globally, right?
Sure.
There used to be an indication where one particular country, I won't mention it, wanted the incubation period to be 30 days. And at that time, it was the USP that was at 7 days for the membrane filtration. So they negotiated, flipped the coin, and went 14 days. Now, what does that tell you? It tells you, though, because the test is designed to pick up a blatant failure. If there's a blatant failure, it's going to grow in 14 days. You're going to see other things besides, a specific odd organism.
Right.
But you can get it, and that's why process validation is most critical. No doubt about that.
So, so I want to talk a little bit about some of the innovations in the area of sterility assurance that you're working on there at Eagle. Can you give us, can you tell us a little bit about that and give us some examples of the impact it's had for maybe some of the clients you're working with?
Well, sure. We have been using here at Eagle the Scanrdi system, which is a non-growth paced system that actually is about, I'm going to say 66% more sensitive than the regular sterility test in a side-by-side test of organisms that grow versus no grow. See, there is a certain class of organisms called viable, not culturable. It means they are alive,
right,
but you can't get them to grow in media.
Okay.
The Scanrdi system picks it up, okay? Now, we use a lot of the Scanrdi system, but by the way, we have the other rapid systems that work, but they're all based in growth. And so if growth base is an issue or something you need or can't for one reason or another be used in a Scanrdi system, we use those growth-based systems. There are several depend upon ATP production or other gas production that you can do and measure in the headspace. But they will miss those that cannot grow. They will miss the ones that can't be cultured. And perhaps that's the best way to talk about them. But the Scanrdi system that we've been using here at Eagle since 2003, 2004 is a system that is not for the faint of heart. It takes some work. It takes some activity. We actually did work and are able now to do oil-based products, which was new, and we have a patent on that. And that was important because many of the hormone replacement therapy compounded products, their shelf lives aren't as long as you'd like to see them.
Sure.
So when you do an oil-based, everyone thinks of the USP 71 test as being 14 days. Well, it isn't for oil. For oil, at the end of 14, because sometimes it looks a bit cloudy when you start it, so you have to subculture. You add another 2 or 3 days. So you're automatically at 16 or 17 days. And if you got 30 or even 45 days shelf life on something, you've just sucked up a whole lot of your stuff and a whole lot of stuff that you're patient for the efficacy of and all that kind of activity. So
right.
we spend a significant period of time because of that product line. Now that opens us up to be able to do other things in other times. But the key is on any test, whether it's a Scanrdi test, whatever test you're using, you need to get the data as quickly as you can so as you can respond to it. Now, we heavily validate all the products that we use in our Scanrdi system, every product, every formulation. Scanrdi system has a DMF, has a drug master file at the FDA, which means that the manufacturer has submitted data to indicate its performance in the hands of the FDA. That's a big deal for us. We like to know that that's there. We also know what's there, and so
right.
we work around it and put it in place. I think it's real critical to know that the USP 71 test is, I don't want to say this like leeches in a barbershop in the 1800s, but it's old. You put something
right.
and you see if it grows. But it works. You have to be able to pass it, pure and simple. So I believe in it. There's no doubt about that.
Sure,
So,
it makes sense. And it's great that you have, some more modern versions of this this tried and true test, right? I don't know, but in this, talking about testing, the sterility testing and everything, it's obvious that we can understand from it that sterility test failures then may not identify the source of the problem. We talked about unculturable organisms, et cetera. And so I know that it's recommended that you have an environmental monitoring process as part of what you're doing. So how can compounding facilities, in your opinion, improve their environmental monitoring program and so they're not just relying on the sterility testing alone?
Well, if it was up to me, I would do more environmental testing than less sterility testing.
Right.
Because I think if you're in a good process, a good environment, a good place, the chance of contaminating is significantly less. I think some of the issues that, and I know not everyone will like what I'm about to say, but the concept of doing environmental monitoring every 6 months or even less than more than that, it's insufficient. If I know what happened in January and July, I don't know what that happened in March.
Right.
And so I think that the real reality is that you have to have a program that measures and can pick up from a sensitivity perspective changes in your environment as they occur. We talked to several people and help them walk through when they have a sterility test positive. We always ask them what their environmental plate said at that time. Those that said, well, I did them according to the USP and I did it 6 months ago, I'm sorry, we can't help you. It means your product was contaminated, and that's where it came from, right? And so at the end of the day, what does that mean? When I said earlier that it's a human being that causes the issue, I don't mean the natural flora of the human being, though it can be.
Right.
We had a situation where we were able to analyze and determine where product was being contaminated in the clean room, making product, because the compounders were walking through an area where there was cardboard and dust that was loaded with mold. And it was that mold that was getting into the product. Okay, and so we were able to identify genetically that mold into that product. How come we looked out? How was that lucky? Because when we got there, they had no data. So we watched around and saw what they did and took samples outside of their clean room. It was outside the clean room, not the clean room. So we could find out, we could determine it was the same genus and species, really a weird one, that showed up in the product. So they moved it all out, cleaned it all out, and life was good again. They do now test weekly as opposed to any other day. But environmental monitoring, remember that the person's the environment. And to me, environmental monitoring always included fingertips.
Right.
Fingertips.
I mean, and you definitely have to have, I would think, like we do in many other areas of drug manufacturing, processes in place to really make sure, processes in place, and I'm talking about for environmental monitoring, to be sure you're doing it with frequency and accuracy, right? And I mean, talk a little bit more, if you can, about where maybe some of the failures in environmental monitoring can come in and steps that people can take to,
sure.
Eliminate that as much as possible.
We've got about an hour and a half. So the fact is, one of the most important things is with any data that you collect is you actually look at it.
Right.
And you have to be careful of the question that you're asking to make sure it's well defined. If you have your best person do all your environmental isolates all the time on the same day at the same time, you've already skewed your data. You need to be testing the environment with the presence of the people who are in it on a random basis, doing different drugs and different products, whether dosage form it is. So at the end of the day, you need to make sure that you have a control. And what is the control? I don't mean negative and positive controls. I mean control of the selection. Do not bias where you took your counts. Do not bias who took your counts. Do not bias the time. Well, I like to do it in the morning because we just cleaned. Probably not a very good thing to do, but I would recommend. Right? I suggest you do it before and after cleaning, frankly. And at the end of the day, the more data you have, the better it is. But to have too much data doesn't count either. You need appropriate data. I think that if you would ask me what is a minimal thing to do, you always know that the individuals, the technicians that are performing the task, not the task of environmental monitoring, the task of producing the product, they themselves are tested or do the testing of the environment and themselves during the process. And they do that across dosage forms, time of day, and week. Now, that doesn't mean you do them all on the same day. In fact, I tell you no. Right. Take a sample on a Monday one day, the following week you do it on a Wednesday or Tuesday, then it's a Thursday the following week. Collect data over time, over seasons. Now, in some places, I'm in Texas, it's always hot and humid. So we can grow anything, anytime, any place.
Right.
People got cold last night, it was 72. So at the end of the day, you need to know your environment. You need to know what goes on. I know that in Southern California, when the Santa Ana winds are hitting, I know that there's going to be mold every place, and I have to be more judicious with my processes. And as such, I make sure I take environmental samples to respond to that environmental condition, which impacts me. I also have got to tell you that environment, we always said we have to talk about people and their environment. If you're coming from home and you have birds, you might have bird dander and bird contamination on your body. We have picked up bird contamination in product, okay?
Oh, wow.
In product, because people washed their hands and everything was good, etc. But they forgot their eyebrows, I guess. I'm not quite sure where it came from, though I must admit I just read a 483 on a pharmacy who said that the technician's eyebrows were in the hood and they were not covered. So I'm not sure if he knows the same case that I know, but I do know in fact that it's that unique condition that doesn't happen all the time that you get zinged with, right?
Right, of course.
So your environmental program should tell you that routinely, in a routine state, in a routine operation, that you are good. But you need to watch for those blips. And the blitz is your own process.
Right. No, that makes perfect sense. It sounds like, especially if you're in a facility that has many compounders in it, that you're going to be doing a lot more data collection, right?
Yes, absolutely.
I'm just trying to understand, like, how often is an individual compounder checking their environment? How many times a day, how many times a week, how many times a month, whatever may apply. Because it seems like you could spend a fair amount of time doing that.
Are you asking me a regulatory question or a science question?
Not from a regulatory, but just from your experience, what gives you the best data, I guess. Okay. So I'm asking you to ballpark, like, how,
sure.
Is it one time a week? Is it 10 times a week? And I'm just trying to, because I'm trying to understand really, realistically, how much time should be spent on this for each individual working in that space.
Plates cost about $2, okay? And we actually sell plates at Eagle. We got a group, we bring them in, we growth test them, so we don't just use the C of A, and we make them available to our clients. And they're room temperature storage. Sure. It doesn't take up refrigerators and stuff like that. If they ask me that question, I ask them, how often do you compound? They say, we compound 3 days a week. Then I said, well, why don't you go ahead and spend $6 and do 3 fingertips every time you do a compound, and know that when you are at that business work that you make sure that happens. And then on a weekly basis, perhaps you select it. You tell me, take an environmental plate, an air sample, whether it's in your hood, which should be just fine, or out in the ISO 7 buffer room. Okay, if it doesn't get into the ISO 7 buffer room with that hood, it's not getting into the hood. So if an air sample isn't doing that, otherwise it's on your fingers.
Right.
So I think I just spent $8.
Okay. Yeah, and again, it's not even about the money. I'm just thinking, like, if you have 8 different compounders working in that facility 3 days a week, 4 days a week, are they each doing a fingertip test?
I think you rotate that. I think if you're a real first of all, if you're a real 503 with that many compounders, you're producing a whole lot of stuff. Sure. That means you have more than one hood. No, but Stacey, it's real because some are part-timers.
Yes.
Okay, and a part-time person perhaps needs to be tested even more because they don't get used to the flow. Right. They don't get used to the run. But if you focus everything on a hood and an ISO 7 buffer room, I got the 8s and the antis, I got that stuff. But if you focus on that, is that where the business goes on, then the people coming in are the ones that you make sure are comfortable with what they're doing and how that works. But boy, I am telling you, once you collect this data, you have to look at it. You have to analyze it. And you have to know that if you get counts that are, quote, acceptable, counts all the time that are acceptable counts are unacceptable from a frequency base. Right. I don't know that many people look at USP 1116 chapter where it talks about the frequency of acceptable limits. Okay. They're not acceptable every day.
Right.
Every time, every shot. So it's how do you look at the data?
Right.
And we'll get calls from our clients and say, I just got this. I say, yeah, nope, that's fine, don't worry about it. How about this? Whoa, you ought to go back and take a look at that one. Because the biggest issues we get is pharmacies use a lot of alcohol. Well, alcohol doesn't kill spores.
Right.
In fact, spores are stable for years in alcohol. Years.
Exactly.
And I want to tell you about 70% of the isolates that we find that are real contaminants are spores.
Which makes sense because they're not being caught there in the alcohol, right?
Right.
So with that being said, then obviously we just said, you collect the data, they need to analyze the data. What tools and methodologies really need to be in place to accurately analyze data that is collected through environmental monitoring practices?
Well, an Excel spreadsheet 101 works just fine, and plotting your data by person, by dosage form. Please write this down, by dosage form, because some dosages are harder, forms are harder to make than others. If you're making a sterile cream or an ointment, trust me, that's not easy. And as such, you will find that everybody across the entire pharmacy will have a problem with it. So that's a process thing. Back to your very good statement about process validation, it gets back right to their states, because their process needs enhanced, all right, or perhaps upgraded.
Sure.
Should be reviewed. So, but absolutely, I think what you have to do is, but you also have to look at, we identify pretty much everything. And because by identifying the organism, we can determine exactly so much information. Is it a mold, or is it an aerobic type of environment that we'll find in the air? Is it from a dust particle or dirt? Is it from animal based or human based? You can tell a lot by the ID. And I know that most most pharmacists will then go to Google or Wikipedia and say, well, this thing was named from something that came from Egypt. I haven't been to Egypt. Well, that's not where it actually came from. So I'm not saying it didn't, but at the end of the day, it tells us a lot. Gram positive versus gram negative, whether it's a mold or not, it's a big deal. It tells us things.
Sure.
So you have to look at numbers, frequency, and ID.
Good to know. Good to know. And so I'm just thinking, because I think about in pharmacy, whenever I'm thinking about pharmaceuticals in general, I'm thinking about the life cycle and the different organizations that participate in the activities that lead to drug making, whether it's compounding or regular manufacture. And I recognize that they're all sort of in a different they might be in differing degrees of where they are along a process. So in terms of implementing an environmental monitoring program, some may have a super efficient program and they've been doing it forever. And I recognize that some compounders might be at the more of the beginning stages with this, right? And they may not have gotten as far along the path. So with that in mind, I just wanted to see if you had any advice for one or two things that every compounding facility can do today, whether they're sort of somewhere in the beginning or whether, they're much further along in their EM program. One thing or two things that they can do today that might improve their overall results.
Yes, that is a great question. The number one thing they have to do is to make sure that their physical equipment, the primary engineering controls, hood, room, pressure rates, and flows are always certified and working properly. Do not make excuses for a failed result. Don't do that. The second thing is your individuals have to be trained how to function in and around a clean room environment. You cannot be blowing up bombs outside the clean room door thinking that doesn't impact it.
Right.
All right. And by blowing up bombs, I mean a microbiological event. I'm popping up cardboard boxes and I'm doing all this kind of activity. And then somebody walks right through there to go into the clean room next to it. Some pharmacies are very small and space constrained. That makes it difficult to keep it clean. Okay. And I think that if you do those two things, make sure you know what you're doing. And I am just a big believer in the fingertip plate is worth the price of gold. And the ones that will tell you that, oh my God, it mattered. They thought they were good, and never failed a sterility test. They did a couple fingertips, and like every second or third one failed, bothered them immensely. It made them think more, to watch what they were doing and be concerned. It's sort of like getting comfortable with the bomb.
Right.
That is something you have to maintain your desire and your dedication and the discipline to do things that you cannot see.
And that's what makes these tests so important, because, sometimes it does highlight things, issues that you didn't know were there, wouldn't suspect were there. And it is very telling. And even though your clients might have been disturbed by that, I think that that's a really good thing for them to have gone through, right? Because now they can account for that going forward.
Absolutely agree.
Yeah. Great. Well, we've covered a lot about sterility testing and environmental monitoring today. And just as we wrap up, Dr. Caputo, I wanted to see if you had any last thoughts or suggestions that we haven't yet covered that you'd like to share with our listeners.
I have several, but I want to say something that's near and dear.
Fantastic.
We are in a very different environment today with COVID.
Yes.
We have a heightened awareness and experience of viruses, contamination in general. Individuals who have the opportunity to perhaps use some of the emergency use applications to go to a non-sterile this or a non-sterile that means that you have to be more diligent. It is not going to change. It is not going to get any better before it gets a touch worse. And I'm only talking about emotional.
Right.
It doesn't matter. It doesn't matter what happens. But please, I beg you, make sure, because you're already supposed to be doing it, if you have the sniffles, go home.
Mm-hmm.
Please do that.
Fair.
So many people say, here's the classic one. I have allergies. That's
Yeah.
okay. It's allergies. Have allergies next year. Go ahead and take care of yourself. What we're doing now for sterility and keeping open the medicines that are needed for individuals, et cetera, we must be diligent, but we must focus on making sure we're there to help the world. That's what we're supposed to do.
That's a great point. And not just for compounders, but it's important for the general public to hear, because I think there's a lot of fear right now from people in terms of, corners that may be being cut or things that may be being rushed along, and people are not quite sure how to react to that. So I think your statement is very comforting.
You got it.
Great. Well, thank you very much for spending time with us today. It's been great talking with you. I really appreciate it. And I'm sure our listeners do too. And we'll make sure that we include some links to Eagle Analytical in our show notes so that people can check out some of the different testing options that you are supplying for some of your clients. I think some of them are very modern, a very modern take, or very novel in the industry. So I'm sure folks would love to look that up.
There you go. I love it. Thank you so very much. I appreciate it. Thank you for the opportunity.
Absolutely. Also want to give a big thank you to Ben Kitchen and the Studio 21 Podcast Cafe. And lastly, a big shout out to you, our valued listeners. If you have topic requests or guest suggestions, we'd love to hear from you about that. And if you enjoyed this episode, we ask you to subscribe in your podcast player of choice. And don't forget to share with your colleagues, your friends, and your online networks. That's how we get more great information out to the masses. For today's show notes and information on the Voices of Validation podcast, please visit www.ivtnetwork.com. We'll be back again next week with another great interview. Until then, stay healthy and safe.
The views and opinions expressed by the hosts, guests, or callers of this program do not necessarily reflect the opinions of the Studio 21 Podcast Cafe, the United Podcast Network, its partners, or affiliates.